Ekis, 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 ekis. From the Llano Estacado to the Rio Grande Valley, from the bayou to the bottomlands, from the desert to the rolling plains, this is a seminar for a wide range. On today's episode of the Soil and Crop Sciences Hour of Power, we have our first student speakers giving their presentations of their research data. Today's speakers come from the barren wastelands and great ice storm producing region of North Texas. With that, I'd like to turn it over to our introducers who can introduce our first student speaker.
Okay, hello. My pleasure to introduce Jennifer MacMillan. So Jennifer comes to us originally from Canada, but she grew up in a Canadian military family. So she's actually born in the United States in Washington, but also lived in Alaska, New York, and Germany. She made her way down to Texas and joined my program. And she's been a really good steward of 3 research projects that she's going to describe to you now.
Thank you. All right. So let me share my screen. Hide that. All right. So yeah, thank you very much, Dr. Finlayson and Dr. Adams. Welcome, everyone. And so the topic of my thesis was evaluating factors affecting biological nitrogen fixation in legumes that are produced in the Southern Great Plains. So to start us off, we'll look at the nitrogen cycle and in particular, the role that legumes play. Nitrogen is typically the most limiting nutrient in agronomic systems, and commercial nitrogen fertilizer is really heavily relied upon. And so looking at legumes can be advantageous in agronomic systems for their contribution to nutrient management. In particular, because of their ability to fix atmospheric nitrogen through a process known as biological nitrogen fixation, or BNF. And so this is a symbiotic relationship with root-associated bacteria called rhizobia, where they fix atmospheric nitrogen into a form that's available to plants in exchange for some carbohydrates. And this is the general theme of my thesis. And I have 3 chapters, 3 projects, as Dr. Adams said, that I'll be going over today. The first is looking at BNF in winter legumes, and the second and third are on a legume known as Guar. The first of those 2 studies is looking at an inoculant product for this crop, and the second is looking at the root system architecture and nodule development across space and time. So rolling right into chapter 1, there is very little information on BNF capacity and the nitrogen fixation potential of winter or cool season legumes in this region. In particular, there's no information on BNF capacity or percent of nitrogen derived from the atmosphere in different species, nor is there any information across different cropping systems. So that's the main objective of this first project, was to directly quantify BNF in 4 winter legume species in 3 different systems. So the main species we looked at were Austrian winter pea, lentil, crimson clover, and hairy vetch. And these species are either commonly produced here or of interest to growers. And the 3 study sites were 3 different cropping systems, all of which were experimental dry land, all managed by Texas A&M AgriLife Research Station here in Vernon, and all of which the plots within each study were arranged in randomized complete block designs. So the first 2 sites are maintained by Dr. DeLon here in Vernon, the first one of which was established in 2011, where they're evaluating conventional and no-till, as well as cover and no-cover crop treatments. The cotton is the summer cash crop, and the winter cover crop is comprised of pea, vetch, clover, and rye. So I'll refer to this one as either Site 1 or the long-term cotton system study. Study number 2 was established in 2018, again managed by Dr. DeLon, and they're evaluating different cover crop seeding rates. Cotton again is the summer cash crop, and the winter cover crops are pea, vetch, lentil, and rye. So this one will either be called Site 2 or the shorter term cotton system study. And then Site 3 is a little different. It is a transitional organic cropping system. So it was in year 2 of transition. And it's comprised of a rotation of dual purpose wheat and a legume-based hay crop. And the legume mixture is of pea and vetch. So Site 3 will be termed the organic system study. So as far as methods go, in April of 2020, above ground biomass was sampled, dried, weighed, and then I processed those in College Station for 15N content at the isotope lab down on main campus. And so we used the natural abundance method in this study. And this method is useful when you're comparing legumes, when you're evaluating legumes that are grown in the same location as non-legumes, and the non-legumes act as what's called a reference plant. And so at sites 1 and 2, the cotton studies, rye was the reference plant grown at the same time as the legumes. And at site 3, the organic site, winter wheat was the reference plant. Now the B value in this calculation is a correction factor. So it accounts for the 15N content of the legume when it's grown on complete dependence of atmospheric nitrogen. So we got those from the literature. So factoring all that into this equation and the 15N we got from the isotope analysis from the lab, we get percent NDFA, which is the percent of nitrogen derived from the atmosphere. In addition to that calculation, we also calculated total biomass, nitrogen content, total assimilated nitrogen, total nitrogen, and then total BNF. And so looking at those, this is for site 1. There's a graphical representation on the right for biomass for the legumes. So pea and vetch had the highest biomass and much greater than clover. For percent NDFA, pea had the highest, followed by vetch and clover. And then for total BNF, again, pea and vetch had the highest and much greater than clover. So now moving on to site 2, again, the legume biomass was highest in pea and vetch, this time followed by lentil. Percent NDFA, highest in pea and lentil, followed by vetch. And finally, total BNF highest in pea, and then followed by vetch and lentil, which did not differ from one another. And then looking at site 3, the organic site, so percent NDFA here was much greater, so almost 93% of nitrogen was derived from the atmosphere. And this may be due to a very low total available nitrogen in the soil in this system, as well as the intercropped winter wheat grown with the mixture of legumes, sequestering nitrogen while the legumes are growing. But then averaging out with the biomass to give you total BNF was comparable to those other 2 sites. So to conclude for chapter 1, it appears that pea is the strongest nitrogen fixing legume, followed by vetch and lentil. Clover did not perform very well here at a very low biomass. At the sites, though we can't compare statistically across sites, they were similar despite there being different environmental and management factors. And in the big picture, this is useful for providing knowledge on the BNF potential of different species, as well as at different systems and different management practices. So this will be helpful for producers of legumes so that they can grow them as cash and cover crops, just more information for their toolbox. So now moving to chapters 2 and 3, these are both about a legume called guar. And that legume might not be well known to everyone, so I'll give you a little background information on it. So getting to know guar a little bit, it is a drought-tolerant summer legume that is grown in semi-arid regions around the world. It's mostly produced in India and Pakistan, but there is some production in the States as well as Australia. It's typically used as a protein, like as a forage for animals, or fresh pods for human consumption, or as there's a guar gum within the seed endosperm. And there's a lot of industrial uses for guar gum. You'll probably see it on a lot of labels for food, as well as it's an emulsifier and thickener in many products that we see, textile products, cosmetics. Like I said, it's a thickener in sauces and gravies. And my personal favorite, it's a thickener in ice cream and some other dairy products. So guar in capacity with an inoculant study. So before doing this research, there was no known commercial inoculant for guar. And I also want to mention that typical inoculants such as the soybean inoculant or cowpea or peanut, they're not compatible with guar. So it's a unique species that needs a unique rhizobia strain. However, the USDA germplasm collection has 4 strains of rhizobia that are known to be compatible with guar. And so we had a great microbiologist help us create a peat-based powder inoculant out of those 4 strains, as well as a mixture of those 4 strains for a total of 5 inoculants that we wanted to test. And so that was our objective, was to test the efficacy of those inoculants on nodulation, nitrogen assimilation, and plant productivity of guar relative to that uninoculated control. To do so, we inoculated guar seed and planted them in 2-gallon pots. So for each treatment, there were 4 replications of each treatment, and we grew it for 50 days in the greenhouse, and we completed this study twice last summer. And so at deconstruction at 50 days, above ground biomass was collected, and then I used elemental analysis to sample for percent carbon and nitrogen. And then below ground, you see here, we sifted through the soil to find all of the nodules. And so this is just a sneak peek at what nodules look like in guar. They range in size and shape. They're typically at the top, kind of 15 centimeters of that taproot, but we'll get more into that in the 3rd study. But we weighed and counted and measured all of the nodules. And then so getting into the results, comparing these inoculants to the control for above ground biomass, these 2 inoculants increased biomass relative to the control. And now looking below ground, nodule weight, which is a good indicator for BNF capacity. 4 of the inoculants increased nodule weight relative to the control. And then total assimilated nitrogen, the same original 2 inoculants increased assimilated nitrogen relative to the control. And finally, protein production, those same 2 were significantly increased in protein production relative to the control. So overall, it appears that those 2 USDA rhizobia strains, number 3089 and 3386, had the most positive impact on guar due to those productivity variables. And those 2 strains may have been more competitive than the native rhizobia in the soil, which is clearly evident by the control also being having successful nodulation. But more research is needed, especially in field trials, and then more research and funding to commercialize an inoculant product for this crop. Okay, so that's number 2. And chapter 3 is really exciting. So up to this point, there's been no characterization of the guar below ground root system architecture for this crop. There's been recent advancements in above ground characteristics, for example, growth staging, growth stages, but up to this point, no information about timing or hierarchy or development of the root systems, nor any information on nodule development over time and space. And so that was our objective here, was to investigate the spatial and temporal dynamics in the root system, architectural, and nodule development of guar. To do this, we used 130 centimeter tall soil columns, and we filled them with a local sandy soil, and we grew one guar plant per column for 80 days. We deconstructed 3 columns every 20 days, so we had 4 harvest intervals, and then within each column, we had 5 soil depths, so we stratified it by 5 depths. And again, the above ground biomass, the same kind of sampling was done as the last study, but the focus here really is on the below ground information. So we sifted through and pulled out all living roots, a lot of washing, a lot of sifting, and all nodules. And so the roots I processed through WinRhizo. So that's just a scan image right there of the top 15 to 30 centimeters, I believe, for that particular root. But looking at the results for nodules, so here we have nodule number and nodule weight on the y-axis, and then time, so days after planting, on the x-axis. And we repeated this study twice, once last year and once this year. So in 2019, as you can see by the different lines, those are the 5 soil depths within the column. And so there was a clear trend that the most nodule number and highest nodule weight was at the surface, and it decreased with depth. We don't really see that trend in 2020, although we do see that there was much less nodulation going on and fewer, less weight in nodules, but we're not entirely sure why the trend isn't as clear. But one other thing to note about nodules that we took note of was when they began to senesce. So in nodules, you see a red interior, which indicates active biological nitrogen fixation is occurring. And we started seeing that transition to inactive, which turns from a red to like a bluish green. We started seeing that around 60 days after planting. And then now looking at roots, the root length density and root weight on the y-axis, and then again time on the x-axis, a pretty similar trend both years, with root length density and root weight being highest in the surface levels and decreasing with depth, and very minimal root length density and weight at depth. But even the smallest amount of roots can do a lot of work in terms of accessing water in the soil profile and keeping the crop alive during a drought, and even growing through a drought. So a little goes a long way with roots. And we get into that a little bit more here in the root diameter classes, which is really interesting. So there's traditionally 2 classifications for diameter classes. You have fine rooting, which is anything 2 millimeters in diameter or smaller. So you see that in these first 3 columns. In 2019 is the top block with those depths, and then 2020 at the bottom. And then coarse rooting is the other classification, and that's everything that's greater than 2 millimeters in diameter. And so if you look a little bit closer at total fine, so everything that's less than 2 millimeters, over 98-99% of the entire root system is fine rooting. And looking at the orange there, like only 1 to 2% is greater than 2 millimeters in diameter, so the coarse. And that's typically that main tap root that penetrates really, really deep, and the rest is super fine. And out of all of that fine, the 98-99%, the majority of that is extremely fine rooting, 0 to 0.5 millimeters in diameter. So lots of teeny tiny roots. So overall, the main points was that the roots grew deep rapidly. So the taproot reached the bottom of that 130 centimeter soil column within 40 days. And this confirms the crop's ability to access water deep within the soil profile, and probably plays into its drought tolerance. As well, nodules were concentrated towards the surface, and like I said, decreased with depth, but this could impact guar's BNF capacity, since the nodules are typically concentrated towards the top. They're susceptible to drought and heat stress and that kind of thing. So that could impact guar's nodulation. And I don't know if I mentioned this before, but guar is kind of perceived to not be a very good nodulator in the field. And we didn't know if that's because when people are making field observations, they're just looking at the top part of the soil profile right along the surface, or if it really is not that great. But these results kind of show that nodules are all the way through the soil profile. So maybe the BNF, the guar is relying on BNF from those lower nodules. And finally, so these results show the root system architecture, which includes a deep proliferative root system with that single tap root that's into that coarse definition of diameter, and then a lot of fine lateral roots. So interesting information for this not so well-known crop, but we've up here in Vernon really enjoyed working with it and becoming the guar guys. Thank you very much for listening. And I definitely have a lot of people to thank, particularly my advisor, Dr. Adams, my committee, Dr. Rajan and Dr. Trussell. I really enjoyed our research group up here in Vernon, and that wouldn't be the way it is without Philip and Rajan. To Dr. DeLon for his assistance and maintenance of those study sites, and as well as the rest of the staff at Texas AgriLife Research, and the wonderful ladies in Heap 217, as well as friends and family who have made my time at A&M wonderful. So thank you so much for your attention. And if anyone has questions, I'd be happy to answer them.
Great seminar, Jennifer. That was extremely interesting.
Thank you.
Questions?
Thank you very much for your presentation. This is Shakira. I've loved the presentation and the new crop guar. So I'm asking about how much is the inoculum? Is it cheap enough like to meet the cost benefit, if someone is to grow the guar like on a big scale? Because when I saw in some of the results, the control also showed that it was producing like something. So how is the cost benefit and the price?
Good question. So right now there isn't a commercial inoculant available for guar. So as far as cost is concerned, it's clearly not effective enough to make one. There have been inoculants in the past, but the technology's kind of been lost. There is one that is available in Australia, but it's not available here. But that's a huge thing that we're looking into, is like how to make like the kind of the price point or the pros and cons of like the economy of scale, right? On how big guar needs to be before a commercial inoculant is available. And so recently there's been like looking into getting crop insurance for this crop. So that might kind of boost things and get things growing a little bit more. And then there's more likelihood of there being that investment into a commercial inoculant, if that answers your question.
All right, thank you.
Hi, Jennifer. This is Katie Lewis. Great, excellent presentation. I really enjoyed it. Question about the first study. Was there any soil sampling that was conducted to ensure that that wasn't influencing nitrogen fixation?
Yes, we did. So we sampled before. So we looked at nitrate levels and ammonium. And I didn't show that. I had like weather data and soil data. I should have showed that. But yes, we did. So there was pretty low levels of both at all three sites, higher levels of nitrate in Chillicothe, so at sites 1 and 2, and then higher levels of ammonium at the organic study, but still very low.
Okay. And did y'all sample by plot?
We did. We sampled in the, this is a good question, in the no-till, no cover, just as to have like a standard location, if that makes sense.
Very good. Thank you.
Thank you.
Hi, Jennifer. This is Amani. I have a quick question for you. So in your inoculum study, you mentioned that in contrast to the control, the protein content was higher for 2 of your inoculums. And I was wondering what protein content, is it within the plant, within the seeds? Does that benefit the gum production? And how is that beneficial for someone who uses an inoculum versus the control?
So we measured it in, so the above ground was comprised of stem, leaves, and whatever seeds were in the pods at the time. So not necessarily fully mature seeds, but it was the whole plant. And it's the percent of nitrogen from that whole plant. And then we used a protein factor to calculate that total protein production. And as far as for production and for like usefulness, it seems that a couple of the inoculants at least have increased the protein production, which could suggest that those inoculants help make this, so guar, like a better forage, a better high quality protein forage for cattle, for example. I hope that answers your question.
Yes, it does. Thank you.
Also, I don't know if I'm still screen sharing. I can't.
You're still screen sharing.
Oh, okay.
Other questions?
Yeah, this is David Baltensperger. And I had a question on the seeding rates in your winter legume studies. What seeding rate did you use for the vetch and the peas?
Ooh, this is a good question. I would have to check my thesis for like the actual written part. So it changed based on the site, I think. And so one of the sites was, they're looking in particular at seeding rates. So there was a different, I think it ranged from 7 pounds an acre to either 30 or 60, but it changed between species. So there were 4 seeding rates per species for that one site. And I think it was around like 7 for some, 15 for some at the lowest, and then 30 to 60 for some at the highest. But I'd have to get back to you on specifics for vetch and pea.
So the reason that I asked is because you got about 60 pounds of nitrogen, and typically we seed about 60 pounds of those. And so you used a pound of seed to get a pound of nitrogen.
Mm-hmm.
And that seems, last time I was checking the price of those seeds, that doesn't sound like it's going to be very cost effective.
Right. Yeah. So we did find with the second site that I was saying, where they had the 4 seeding rates, we did a analysis of those to see if there was a difference for biomass, and we showed that there was no difference. And so then we kind of grouped it all together and then analyzed it as one within each species, if any of this is making sense. So I think that proves your point, and maybe going with, like, this obviously isn't the point of my research, but going with the lower seeding rates, obviously, to save money could work. But I don't want to make that assumption because that wasn't my
That's fine.
So, but yeah.
Great job. Great work.
Thank you so much.
Other questions?
This is Reagan Noland out at San Angelo. You made a point about possibly having more fixation in a lower nitrogen environment, and that's something we know about legumes. You know, it's an energy expensive relationship for them to trade off those carbohydrates, right? It's kind of a conflict I've run into with growers that I work with that are interested in integrating legumes in their systems, because a lot of these are also the guys who are really tuned into soil fertility, and if there's something lacking, they're pretty ready to fertilize. And so I'm like, well, the legume might not fix as much nitrogen or any at all if you've got adequate soil nitrogen available. And they kind of stop me and say, whoa, you know, what am I doing this for? How does this work? And I want us to be able to take advantage of and rely on legume fixed nitrogen more in our systems. And I like the point you just made about figuring out what is the lowest we can go on our seeding rates to make it the most cost effective and that sort of thing. But do you think that the total amount of nitrogen that we're fixing or contributing is ever going to meet our need, or are we going to have to compromise yield relative to what we're fertilizing, just in totally, fertilizer driven systems right now? I'm not sure how those will compare, but I'd be interested to hear your thoughts on that.
Yeah, I mean, it's definitely a good point, especially it's like the real world scenarios. And I think in time and with research and development and stuff, we could get there. But ultimately yield, right? Yield is king. And so that trade-off is an interesting process. So yeah, I'm not really sure. I think it's just the same conundrum, right? Where we want to push legumes and for their benefits, and people are very interested in soil health, and but if it doesn't,
in a system that's being marketed as value added, like organic, it may be easier to justify or to approach, because that it's harder to come by nitrogen in that system, right? So
for sure,
than adding fertilizer. And so anyway, I appreciate your talk. I think you did a great job. I was just that was something that kind of stuck with me. I was like, man, we got to figure this out.
Yeah, we do.
All right.
More.
All right, we got to move on. So thanks again. It was a very interesting seminar, I must say, and lots of good interaction.